Have a question or comment for the speaker, simply type your question in the chat feature. Liz Chudoba will read as many questions as possible during the Q&A period and allow the presenter to respond. I would like to now introduce our first speaker this afternoon. Dr. Upal Ghosh will present on in-situ PCB PAH remediation using sediment amendments. Dr. Ghosh is a professor in the Department of Chemical, Biochemical, and Environmental Engineering at the University of Maryland, Baltimore County. His group performs research on environmental engineering and science with a focus on the fate, effects, and remediation of toxic pollutants in the environment. His research has contributed to the development of transition of novel sediment remediation technologies. Welcome, Dr. Ghosh.
Thank you, and good afternoon, everyone. And I hope we'll have an opportunity in the future to meet in person. And thank you for giving me this opportunity to talk about our work. And as you can see from the title, we are making a bit of a transition from water quantity and water quality issues to include toxic chemicals in the water. And specifically, I'll be talking about some of the work we have been doing, looking at protecting the water quality and what it supports from effects of toxic chemicals. So in the title, we talk about PCBs, polychlorinated biphenyls, polyaromatic hydrocarbons, but the work that I will be talking about covers a whole range of hydrophobic chemicals that are toxic and bioaccumulative. And these would also include some of the chlorinated pesticides, for example, dioxins, and many other hydrophobic chemicals like that. So the focus here is on remediating contaminated sediments. But before we get into contaminated sediments, it's important to keep the full perspective of these toxic pollutants and how they transport and impact organisms in a water body. It's important to keep that in mind. So a lot of these chemicals, like polychlorinated biphenyls, they were released in the past, they were banned in the 70s, but still continue to impact us. And the reason for that is these chemicals tend to persist for a long period of time. So another important and nasty characteristics of these chemicals is that they are persistent. They don't flow out into the ocean, but stay embedded in the sediment deposits because these chemicals are hydrophobic. They don't like to be in the water phase. They bind to particles and settle out. So in many of these cases, we have contaminated sediments under a water body, and we have many cases in our area here. I do a lot of work in Maryland, in Virginia, so I'll give you a few examples of our work in Virginia as well. And nationally, it just plays out in a big way to impact water quality. Next slide, please. So the water quality impact that I'm talking about are related to bioaccumulation of these chemicals in the aquatic organisms, like the fish you saw in the previous picture. And what it does is it not only impacts the living organisms and the fish-eating animals and birds, but also humans that would go and catch the fish and try to consume it. So nationally, we have lots of fish consumption advisories, many in our states here in Virginia and Maryland, that advise people not to eat the fish or limit the consumption of the fish that are caught locally. So the challenge for us is to figure out where these pollutants are coming from, how they're getting into the fish, and how we can protect the great natural resource we have. The problem is that these sites are large that are contaminated from past activities, and these chemicals have the tendency to, as I said, to bind to suspended particles. And so the legacy of those deposition over many decades when they were released in the past, primarily in the 40s, 50s, 60s, they got deposited in areas where sediments deposit. And oftentimes those end up being wetlands, beautiful wetlands like this one in the picture. This is actually from Fort Eustis, Virginia, where we did a pilot study several years ago. And this is, I believe, Bailey Creek is the name of the creek. And it flows out into the James River, if I'm remembering correctly. And it's a beautiful functioning wetland. And we have brute force techniques that we can use to remediate that wetland, for example, which is contaminated with PCBs in this case. We can dig out that wetland with the contaminated sediments and put it in a landfill so that the contaminants go away. But in the process of doing that, we would extensively damage that existing ecosystem. So if we want to step back and we want to think about what we are really trying to do here, which is reduce the exposure of these chemicals to the aquatic organisms, we're really trying to reduce that exposure and risk associated with these chemicals. We don't necessarily want to take those sediments out. So how do you clean up these ecologically sensitive sites without destroying it? And that was kind of a big motivation that got us started in here. To answer that question, we had to dig a little deeper and think about where the chemicals are and what happens when we try to use these traditional approaches of managing contaminated sediment deposits in water bodies. So next slide, please. So this is a picture. This is a busy slide. Let me take you through this. The one on the top. So both of these, actually, the one on the top comes from a National Research Council study that was done a few years ago, looking at effectiveness of dredging to remove contaminated sediments and the impact on water quality for 20 megasites that were cleaned up. So these megasites are large sites that took more than $50 million to clean up. And what they found was that 50% of those sites that underwent the cleanup with removing the sediments didn't achieve the goals. And that was kind of sad after all that expense, that wasn't quite effective in achieving the risk reduction goal. And at the bottom of that story is the fact that when we try to remove sediments, we create a lot of disruption. So the picture on the top shows PCB concentrations in the water as a function of a dredging activity that happened over a period of time in one of the rivers in New York. And at the bottom left corner of that graph, you see the characteristic baseline concentrations, which were really low. As you start removing sediments from that water body, those levels of PCBs go up because we are starting up old deposits of sediments with PCBs. And once the dredging stops, those water column concentrations fall back to the baseline levels. And that's been a problem. And with using traditional technologies to take care of contaminated sediments. And the reason is the figure in the bottom shows profiles of these legacy contaminants, in this case PCBs in Lake Hartwell. And if you look at the depth profile, so on the y-axis you have different depths in the sediment, the highest concentrations are associated with the time period when polychlorinated biphenyls were released at large numbers, in large quantities back in the 60s or so. And then it has been continually coming down. As we dredge and we remove, we expose some of the more contaminated stuff, which then increases the level of contamination that we have in the water column, at least temporarily. So that's been a big challenge. So a lot of our work went into understanding how these chemicals behave in these water bodies where you have contaminated sediments deposited. Next slide, please. So at the core of this is an understanding of how chemicals go from the sediments into the water column and into the fish, and its absorption of these chemicals into the geochemistry or geochemical components in the sediments that controls that exposure. Some types of sediments tend to bind these pollutants very strongly So these chemicals are not available for uptake into the fish. And in some, they are not bound that strongly, and they are more bioavailable for uptake in the food web. And some of our early work looked into understanding the difference across sites that we were observing in nature, where some sites were holding on to the past legacy more strongly than others. Next slide, please. So at the core of this, again, issue of sorption processes controlling bioaccumulation was a carbon story. So we all hear about carbon in negative light when it's in the atmosphere in the CO2 form. But carbon plays a major role in life processes. And so when we look closely into sediments and geochemical components in sediments, we find a lot of carbon. And there is an interesting link here between the carbon in sediments and these toxic pollutants, because these toxic pollutants being hydrophobic, they tend to like hydrophobic surfaces. And these carbon containing materials in sediments offer hydrophobic surfaces. And there is a whole range of these carbon containing geochemical components in sediments, starting from woody debris, breakdown of plant and animal matter, which is the decaying life forms, and they are hydrophobic and they tend to absorb these hydrophobic chemicals. But in addition to that, we also find another type of carbon which ends up having a very unique relationship with these hydrophobic chemicals. We call them black carbon. So they range from coal, charcoal, some charcoal coming from human activities, some char coming from forest fires. Then we also have soot coming from burning or partial burning of woody and other vegetative material. And when you look closely under a microscope, we find that you have this whole range of organic materials in sediments. When you look at how strongly these chemicals, hydrophobic chemicals, bind to these different components, we find that the black carbon component, the charcoal, the coke, and the coal, have nearly 2-3 orders of magnitude stronger sorption for these chemicals than natural organic materials. So in this early work, we've finally found the reason why some sites tend to bind these chemicals strongly, and we found that those sites tend to have more of this black carbon. So if you can go to the next slide. So we start off with natural organic matter, where carbon is bonded to hydrogen and oxygen, and that's what we are made of. That's what plants are made of. That's what decaying vegetation is made of. And that has some sorption capacity for these hydrophobic chemicals. But when that material goes through a high temperature process, like in a forest fire or in a coke oven, or in my backyard barbecue, I produce this black carbon, which is a graphitic form of carbon. It changes the structure of the surface, making it a lot more sorptive. On this, and these hydrophobic chemicals then bind to the surfaces of these black carbon materials. Industrially, we can take that char, which can be either the biochar, which would be charcoal coming from different organic, different woody materials, to coke that comes from coal. Now, coal is also woody material, just diagenetically altered over millions of years. We end up with this material which has a graphitic surface which absorbs these chemicals. Industrially, we take these materials and we can activate it, which is increase that surface area through a steam activation process and make activated carbon. And many of you are familiar with activated carbon and the use in water treatment, because it has the super high surface area which absorbs these toxic chemicals and allows us to clean air, clean water where we need to. So I wanted to give this the transition going from biomass to biochar to activated carbon, because there is a big carbon story in what I'm going to talk about next. So what we found is that naturally in these sediments where we have this black carbon present, these toxic chemicals are bound up very strongly. So we published several papers in that series looking at that natural abundance in some sites where bioavailability of the toxic chemicals are altered naturally by the presence of these black carbon. We then took that a little further as as engineers. We want to solve a problem once we understand what the problem is. So a big part is understanding and defining the problem. Once we define the problem, we can try to find solutions. So if we go to the next slide. So what we did was, some of the early work we did involved taking contaminated sediments contaminated with PCBs, polychlorinated biphenyls, or PAHs, or DDT, even methyl mercury, and look at how aquatic organisms take up those chemicals, and what can we do in terms of altering that geochemistry by bringing in more black carbon, because in some of these natural environments where we saw less biouptake happening, we saw the role of black carbon in those sediments. Now the question is, can we add black carbon to these sediments, either biochar or activated carbon, which is tailored to have strong sorption capacity for these chemicals, to basically outcompete the organisms in these sediments for these toxic chemicals. So some of the early work looked at beaker and aquaria experiments where we would put a layer of this black carbon material on the surface of the sediments and allow freshwater or saltwater organisms to inhabit those sediments and do the mixing on their own and take that carbon down into their burrows, as you can see in the top second picture, and then take those organisms out and measure those chemicals, in this case PCBs, in the tissue of these organisms. The figure on the bottom right is actually sediments that came from Bailey Creek, Fort Eustis, where we grew this amphipod, Leptochirus plumolosus, and that's an organism that lives in brackish waters in sediments and is at the base of the food web, aquatic food web. And it's a good indicator species in terms of what accumulation is happening in these contaminated sediments. So the red bars are, so the figure there shows PCBs in the organism tissue. The red bars represent the concentration that is uptaken in the tissue by these organisms for the different PCB homologs. So homologs are the different chlorination levels of the PCBs. As you know, polychlorinated biphenyls are 209 different compounds, depending on where and how many chlorines you have on the molecule. So for example, if you look at the tallest bar there, that's a hexachlorobiphenyl. So that PCB that's the sum of all the molecules that have 6 chlorines on the PCB molecule. So the red bar is the untreated sediment, the controlled sediment, and the organism picks up a lot of those PCBs. But if we add 5% by weight of activated carbon into those sediments and then have the organisms grow in that, we get a big reduction in the uptake in the organisms. And basically what is happening is that the PCBs are getting bound in that carbon so tightly that the organisms are finding it difficult to take it up, even though they are ingesting those sediments, even though they are exposed to the pore water that's at equilibrium with those sediments. So we get about, so this is for the second generation. So these organisms grew and produced new ones, and the new ones, after 60-day exposure, they are showing 82% reduced uptake. So that's great, that if the base of the food web can be manipulated with the geochemical manipulation we did in these beakers, that these organisms take up 82% less, that can be a way of interrupting that pathway of exposure to the organisms without actually destroying the ecosystem. So that was really exciting. And we published a bunch of papers based on that. We went on to we moved from these benthic organisms at the base of the food web to, the next slide, please, to the species that we may be more interested in, the fish. And this was a study supported by the National Institutes of Environmental Health Sciences, the Superfund research program, where we looked at the next level in the food web in the fish. And we saw similar reductions. In this case, concentrations in the fish were reduced by 87%. So that was great. It's not only happening at the base of the food web, but also impacting the pelagic fish and these fish are ingesting some of these worms. So they are also breathing the water, and overall exposure is coming down in the fish as well, which is great. Now we can do that in these aquaria settings in the lab. Next slide, please. And we and many others in the scientific community replicated these measurements for a whole range of contaminants, PCBs, PAHs, DDT, dioxins, and the results were kind of similar across many labs, many experiments, many different sediments and different chemicals. And you can see here reductions in biouptake in the range of 70, going up closer to 95% reduction for many organisms. And that was really great. These are results from the lab studies. But can we take this out into the field? How is it going to work in the field? I can do it in a beaker, but do I have a way to engineer that in the field? Next slide, please. So conceptually, the way this might work is that we may be able to amend the surface of contaminated sediments with a carbon amendment that has a very strong sorption capacity for the chemicals that we are targeting. And that would create a layer on the top surface of the sediments, which basically what it does is interrupts that pathway of exposure into the fish. So direct exposure from ingestion of sediments and the sediment living organisms by the fish, which is the left arrow in the bottom there. And also the other pathway of exposure that happens for fish in the water column is through diffusive exchange of the PCBs from the contaminant sediments into the water, uptake into the phytoplankton and the pelagic food web. And we can interrupt both of them by bringing in this additional material that enhances the geochemistry in the right direction, which is making the sediments more adsorptive for those chemicals, so less goes up into the food web. So the question is, how do we engineer this in the field, right? So we worked with several engineers and consultants and people who do sediment remediation to come up with ideas. So next slide, please. There are basically two approaches of doing this. We can add the carbon only to the top surface of the sediments and the organisms mix it up. And we create a bioactive zone which has been altered in its geochemistry to make it more adsorptive for those chemicals. The other, in sites where the sediments are less stable, you can blend it with sand or gravel to create a barrier layer that creates a new habitat which is protected from the contaminated sediments below. So those are the two constructs that we can take out into the field. We had the opportunity to try this out. So next slide, please. We worked with several agencies and people who were trying to find solutions for contaminated sediments without having to dredge those sediments. And we did this across the country and also with several collaborators in Europe and tested it out in pilot scale first. And a lot of these studies basically told us the same story, that if you can get the carbon into the sediments in the bioactive zone, maybe top 4 to 6 inches, it reduces the biouptake of these pollutants in the food web, reduces the transfer of these pollutants into the water column, reduces the concentrations in the water column by enhancing the way it's bound up in the sediments. Next slide, please. So we got some recognition of that work. I published a feature article that came out with a cover page picture of our early work in San Francisco Bay, where we did in a tidal mud we did a pilot study in a tidal mud flat in Hunters Point Naval Shipyard. That was funded by the DoD. And then US EPA came up with a directive on looking at in-situ amendments as a way to remediate contaminated sites. And this came out several years ago. So there has been some acceptance, I would say, in terms of recognizing that this is a viable approach for contaminated areas and sediments, which is impacting water quality. Let me give you a couple of examples before I end. Let's go to the next slide. So one of the first things we did was try out in a river. This was funded by EPA and Alcoa in Grass River, New York. And we brought in the heavy equipment to mix the carbon into the sediments. And it worked, reduced bioaccumulation by 80 to 90% in the benthic organisms. But this was a lot of sediment manipulation in situ, and associated with it was a heavy cost of mobilization. So we had to come up with a different approach to get the carbon into the zone that we are targeting. So next slide, please. So we are fortunate to get some support from EPA through a small business innovative research project, which was then supplemented with additional funding from National Institutes of Health to come up with a novel approach of delivery of these amendments, which has low impact. So one of the problems with carbon is that it's very light and it would float in the water. So we did some material engineering to make these pellets, which are basically carbon, clay, and sand made into these little briquettes that can be applied from the water column, and then it sinks into the bottom of the sediment and slowly over time breaks up, releasing the carbon. And the natural biological activity by the sediment benthic organisms mixes that carbon into the sediment. So one of the first trials we did was at Fort Eustis in the Bailey Creek, and that's the picture you see of me doing that application in a small area from a boat in the wetland. Next slide, please. The other one is work we did in a little bigger area, and this is a 5-acre lake in Dover, and this work was supported by the Delaware Department of Natural Resources. If you click on that, I think there should be a video that here the same materials are being applied in the 5-acre lake using a telebelt. And so you have to scale up, work with engineers to figure out ways to do that amendment. And as you can see in the top right picture, the carbon after a few months shows up in the top 4 inches of the sediment. And that's kind of where we were targeting. The idea is that it traps the contaminants in the sediments and less comes up into the water column and should impact the fish. Let's go to the next slide. So here are the results from that 5-acre lake study. So that's kind of a big aquarium for us. And we studied the reductions in the concentrations of PCBs in the pore water and in the surface water of that lake. It's part of a tidal St. John's River there, I believe. And so it's a tidal system. So there are some migratory species that are going back and forth in the lake, but there are also the resident fish. And we saw about 70% reduction in the resident fish in the lake. So that gives us the confidence that this is working out in a lake system and that those concentrations in the fish are coming down. So let's skip the next slide and go to the one after that. I'm running out of time, I realize here. So there are a few full-scale applications that are coming up. One in Virginia that some of you may have heard of. This is in Paradise Creek. It's part of Elizabeth River, and where the idea is to do partial dredging, capping, and also in-situ treatment with activated carbon that should be coming online by the end of this year. And then on the right is Scandal Reservoir that EPA and this is dioxin contaminated sediments, and 14 acres are going to be treated with carbon amendment into sediments. Let's go to the next slide. So since I'm out of time, I'm going to skip the concluding thoughts. It's kind of a summary of what I just talked about, that this kind of an approach is applicable in depositional areas where the contaminant bioavailability is high and ongoing inputs have been controlled. It's important to control ongoing inputs first. Let's go to the next slide and I'll conclude there with acknowledgements. All this work would not have been possible without the tremendous amount of hard work that a very large number of graduate students, undergraduate students, and postdocs did over the years, and the funding sources and collaboration with multiple people. And I have a disclosure statement in the bottom that's important, and that is I'm also I also I'm a partner in 2 startup companies that are transitioning these technologies out into full-scale and commercial enterprises. Thank you.
Thank you, Dr. Ghosh. It's a very fascinating topic. I remember you giving that presentation when I was at DEQ, and I found it fascinating then, and I still find it fascinating. I think we do have maybe a time for maybe one question, and we may have to alter our agenda a little bit. We're having a little bit of difficulty with our next speaker getting online, I believe. So Liz, question?
Yes. We have one question. Have you investigated the impacts of the black carbon on other ecologically important compounds? In other words, could it limit essential nutrients or minerals?
Oh, that's a very good question, and that's always in the back of our mind. So we have done several studies, and others have done as well. And what I will do is there is a review article that came out a few years ago, which summarizes all of that. So the basic, in a nutshell, what people find is that when you do these studies, some of these studies in the lab, you might see some effects, but when we do them in the field where there is a nutrient supply that is happening and it's an open system, people don't see any negative impacts as long as the carbon dose is below 5%. So if you go to higher doses of carbon, which often we don't need, then you start changing the matrix so significantly it's not sediments anymore. It's starting to look carbon. And then many organisms may not find it habitable from the physical texture of the material, or because nutrients are getting absorbed and some micronutrients are not being available as well. So those effects start manifesting after we increase the dose beyond something like 4 or 5%.
Great. We'll do one more question while we're waiting for Mike to try to get on. How would the coke be treated as it is taken from the coal plant to a contaminated water body?
Could you repeat that again? How's the coal?
How would the coke, C-O-K-E, be treated as it's taken from the coal plant to the contaminated water body?
Yeah, so the activated carbon that we use can come from coal as the raw material, or it can also come from biomass. So if we look at the large availability of activated carbon in the industry, a big part of it comes from nut shells, coconut shells, and other hard nut shells, and those are actually used a lot for water treatment because they have some really good properties and doesn't have any of the legacy contaminants, heavy metals that you might see in coal. But if it is used, if the carbon comes from coal, which is done in many of these sites, one has to be very careful about the source of the coal and make sure that don't have heavy metals in the material that's being added. And there are ways to get any heavy metals, if they are present in coal, out of it. But that's absolutely important to make sure you're not creating a problem.
Sounds good. Thank you.
Great. Thank you very much, Dr. Ghosh, for the presentation. I believe we have our next presenter ready. We're just having a little bit of technical difficulties. So I believe our next speaker, Mike Hayslett, is on the phone. And he is here to talk about vernal pools. Mike is the principal consultant for Virginia Vernal Pools, LLC. In his role, Mike consults for and educates, organizes, and organizations and private landowners regarding vernal pool management and ecology. Mike has studied vernal pools since his childhood in Virginia Western Highlands. He has written many articles on the subject and traveled all over Virginia educating others about vernal pools. Welcome, Mike. Mike, can you hear us?
Hey Mike, I believe if you hit *6 on your phone, you should be able to unmute yourself.
Hello, can you hear me? Can you hear me now? Hello?
Yes, we can.
Oh, wonderful, miracles. Okay, you can hear me fine, Matt? Hello, Matt, can you hear me okay? I'm sorry, I don't know if we have audio connection through the phone.
We do. We can hear you, at least I can hear you, and we have your presentation up and running.
Very good, then I'm going to proceed. Matt, if you're on the other end at the title slide, I will say next to proceed. First of all, assuming everyone who can hear me, sorry for the difficulties, thank you very much for the invitation. Always delighted to talk about my favorite subject. So hold on to your hats. I'm going to give you the fastest, most succinct talk on vernal pool ecology and conservation in the history of the universe. Next slide, please. I would like to dedicate the talk today to the memory of my father. This is the 10th anniversary of his passing, and spent many wonderful days afield with my dad, exploring vernal pools and other wilds here in the Allegheny Highlands. So I appreciate that indulgence. Next. So vernal pools, to some folks, is a brand new topic. I have spent, my entire career and all of my adult life pursuing these types of habitats. I have studied, I have advocated for their conservation, I have built some, I have restored others, trained many people, and consulted in this arena as I continue to do today, and gained a great deal of satisfaction out of the pursuit of conserving these unique habitats. Next. I'll start here with a little snicker and a grin. This is my first selfie on a project in Northern Virginia almost a decade ago, and that is a constructed vernal pool, what I like to call a facsimile. And go to the next slide, please. And here is that same site when I visited this spring, almost a decade after the construction of that vernal pool facsimile. And I show you this just to insert why vernal pools are appropriate to the overall topic of the conference today of water monitoring. Monitoring is the means by which those of us on the periphery who have come to know these little wetland systems intimately, monitoring using basic observational science is how we have come to understand the ecological importance of these systems, how they function. Monitoring is also how, in this case, in trying to create or restore, monitoring is obviously how we determine whether our efforts are working, if they're being effective. So monitoring a created wetland is the way we determine if it is meeting the objectives that were set forth. And monitoring is particularly important now in the face of climate change to understand what the future of these habitats may be. Next. So again, wetlands generally are endearing habitats to those of us who are natural history or conservation types. Wetlands are fascinating environments, biologically rich, a lot of fun to explore. And so again, over the years, I have found them to be very interesting living laboratories and a great educational outlet for people interested in conservation biology. Next. So the slide before you, I trust, says a plethora of pools. One of the things that has driven me for many years is the variety by which these so-called vernal pools exist in contexts across Virginia. They can vary from dunal swales along the Atlantic coast beaches to floodplain pools in the major river systems across Virginia, particularly in the Piedmont. And in the western mountains of Virginia, both the Blue Ridge and the Alleghenies, where I live in the western highlands, there are upland wetland sites that are fascinating. So this is a wetland reference that we use for vernal pools that spans a wide variety of wetland categories, and they reflect really the variety of environmental conditions across Virginia. But that's part of the fascination. No two are alike. Next. So let's try to just very briefly define what a vernal pool is by using a sort of run-on adjective descriptor. So these, of course, are non-tidal or freshwater wetlands, interior wetlands, and they are generally isolated from other water sources, not typically connected to flowing water. They do tend to be forested, as our part of the world was historically mostly a forested landscape. Probably most crucial is they are seasonal. That is to say, the hydrology is such that these ponds or pools or wetlands hold seasonal water on the surface. And these images here show a site in central Virginia, both during the wet phase on the left and during its dry phase on the right. So this is quite the juxtaposition of an environment that is both aquatic for part of the year and then terrestrial for another part. I like to think of them as a double dose of biological diversity because of these contrasting environmental conditions. Next. Again, here's another image of this contrast. You can see this little pond, and this is on a ridge top, 3 blocks from my home, and this is where it all began for me. This was the vernal pool that I explored as a boy here in the Allegheny Highlands. And you can see the difference there. I was fascinated by this. A site that could be a pond for part of the year, and then during the summer would be this open glen, and I could turn over those sandstone boulders and find ringneck snakes. So the contrast in these conditions just fascinated me from the very beginning. Next. I'd like to tell you a little bit about what I have spent the last 4 years doing, and that is a study of vernal pools and related wetlands and unrelated wetlands in my region here. So our national forests in the western highlands here encompass more than 250,000 acres across parts of 5 counties in the Allegheny Highlands or Western Highlands region at the headwaters of the James River. And I've spent the last 4 years seeking out and inventorying many of these wetland resources as the dot map shows. Next slide, please. So I'll not labor this, but to say a project for the U.S. Forest Service, a special survey project, and I looked at all of the water features found on the National Forest lands in my region, and these were divided into 3 basic categories. So naturally occurring seasonal ponds, or what we call vernal pools, vernal ponds, artificially created or constructed water holes, which are the opposite, and then a miscellaneous category that includes things like seeps and springs, etc. So over the course of that work, more than 234 sites investigated, assessed, geospatially inventoried so that the agency could have this information to manage these habitat resources. And it's interesting to note that just a little under a quarter of all of these were in fact these natural vernal pools or vernal ponds. And that's a pretty significant rate of occurrence over here in our mountainous region. Next. So let me show you some examples. Here is a vernal pool. This one's pretty big. This is about a quarter acre. It's only 18 inches deep. And this is in Bath County. This is a really nice quality vernal pool that has a sort of semi-short cycle of ponded water on the surface. Next. And here's the contrast. This is a constructed water hole. This was built, you can see in the background of this image, the dam or the berm with an outflow pipe. And this is more than a half acre. It's exposed, it's quite deep, and it has the usual sort of artificial pond flora and fauna. There's cattails and yellow flowering spatterdock. And these tended to be built in those days for game species benefit, so to attract ducks, waterfowl for hunting. And they are often stocked with fish. And that, of course, is a major contrast between an artificial pond and a vernal pond. No fish in vernal ponds. Next. So the final category, the miscellaneous, the catch-all, included mostly natural but flowing wetland systems like springs and seeps. These would be littoral or flowing systems, not isolated wetlands like vernal pools. Next. All right, so if you're at the slide with the apples and the oranges, this was really, in addition to providing the agency with the basic information to be able to manage their resources, this was a personal objective of mine, was to clearly communicate the differences between these artificial versus natural wetlands in the defense of my beloved vernal pools. So you'll note the natural seasonal pond or vernal pond is seasonal because it doesn't have permanent water, it doesn't support fish. And fish are the primary predator of some of the wildlife that specialize in vernal pool utilization. So the last item, the function, specialized species are able to use vernal pools in addition to the general fauna in particular that can utilize a vernal pool. I'll trust that you can get back to my presentation later. I do have links in here so you can pursue further info. But next slide, please. Thank you, Matt. So the wildlife water hole, and there are 3 historic photos here on the desk. This is a relic, really, of the post-World War II days of cooperative wildlife management. Animals were stocked, brought from distant places. We don't do that anymore. And these water holes were constructed. And they do have general function in the landscape for wildlife, but they are not vernal pools. Next. So back to apples and oranges. Again, a vernal pool can accommodate the generalists, but it specifically can provide the need for specialized flora and fauna. So they're very often uncommon, rare, even threatened and endangered species that are found in these isolated wetlands, these insular populations that have been around for a long time. Look at the age here. Constructed water holes are less than a century old in most cases, but some of the vernal pools have been shown to date back to the last ice age. So that clearly means that in terms of management, these historic sites really should be afforded an absolute priority in managing them as natural area preserves, where the other things can be manipulated here and there according to the wildlife objective. Next slide. Here's another example, and this is at a particular area here in western Allegheny County that is really fascinating. It is it's a karst or limestone underlain landscape. And let me tell you a little bit about it as I further develop here the ecology and conservation of vernal pools. So this site is up on the mountain. It is a terrace along the mountain created by limestone dissolving underneath and this collapsing and creating a depression along the slope of the mountain. And this site, when I surveyed it, when I assessed it, it was so beautiful, verdant, and rich in vegetational growth during the growing season that I kind of broke from tradition and actually named it something a little cheeky based on a cartoon. But this environment was so beautiful and exotic in its plant community that I called it, yes, Fern Gully Swamp. Next slide, please. Speaking of plants, these environments are very important places for native plants, and particularly for highly specialized and even rare plant species. Here are some of the more common things. If we can go to the next slide, please. This slide should read Bulrush Pond, and this is one of the sinkhole ponds or karst wetlands in that Castile Sinks area. And this particular pond is so named because it harbors the federally endangered plant, the northeastern bulrush. So there are often rare species, rare elements. And I should remind you that these ecosystems, not just the rare elements of plants and animals, but the system itself or the community, these are regarded as globally rare natural communities by our state's Natural Heritage Program. Next, please. All right, so your slide should show Castile Sinks. And this is sort of a picture of that whole neighborhood. And if you will note, there are 1, 2, 3, 4, 5, there are at least 5 ponds. These are sinkhole ponds or vernal ponds. And in addition, there is a cluster of springs that also occur here. So this is a rather complex area because of the unique geology underlying it with underlying limestone, where there are these fascinating wetlands, including vernal ponds. Next slide, please. Okay, here is a picture of all kinds of critters, and I am a critter guy. I am trained as a zoologist. So these are the animals that have been my gateway studying the fauna of these sites. Now, these are all generalist species, and you could see anything here on this list in or around vernal pools in any given season. Next, but there is also a suite of very specialized fauna. These are animals that we refer to as obligate species. They are indicators of the unique wetland that we call a vernal pool. And these include, in Virginia, 6 species of the mole salamander family, several anurans, and then several species of a freshwater crustacean. Next, please. So this slide says spring fairy shrimp. Now these are small, freshwater, and rather bizarre in appearance shrimp that exist in these vernal pools in a single generation life cycle strategy. They're typically winter. We also have a species in the summer. Next slide, please. But I would encourage you, go to this link, this Vimeo documentary when you have a chance. A friend of mine produced this. It's coming up on its 10-year anniversary. It's only 30 minutes, but it's a wonderful overview of vernal pools through the eye stalks of fairy shrimp. It's a great little watch. Please check that out. Next. In addition to the fascinating biota, plants and animals, again, an emphasis on the fact that these are very historic, very old environments. This site is in the central Blue Ridge Mountains on a low ridge top, and in addition to it being a really cool vernal pool with 4 different obligate species of animals, it is a great archaeological site. The artifacts you see in the foreground there, right, bottom right, those are archaic remains of Native Americans when they encamped around this seasonal pond some 4,000 or 5,000 years ago and manufactured stone tools while they were encamped there. I'm convinced that these ancient peoples were hunting ducks and deer routinely at these environments in different seasons. Next slide, please. I'll tell you very quickly about Brown's Pond in Bath County. Now, this is a rather large sinkhole wetland in the upper elevations of the Allegheny Mountains here, and it is a particularly wonderful showcase of ancient history. Next slide, please. This image of Brown's Pond shows it during the growing season, and you can see cinnamon fern in the foreground and three-way sedge throughout it. In recent years, I've been collaborating with a research team of the U.S. Geological Survey, and they have repeated soil core studies of this pond, and the pollen record there confirms that it is more than 17,000 years old. Our karst wetlands in the Allegheny uplands here were formed after the last ice age. So these really represent some of the oldest, unchanged showcases of biological diversity that remain in our landscape today. Next slide, please. My apologies for the neighbor mowing their lawn. I hope that doesn't disturb our talk. So this slide shows a very rare ecosystem that we would think of as an oversized vernal pool. In the upper Shenandoah Valley, the sinkhole ponds there are renowned as being these showcases of relict and disjunct plant and animal species that have been there for a very long time. This area in particular is on the National Forest. There are some 36 on perhaps 400 acres, and the suite of rare elements there is something like 24 rare plants and nearly a dozen rare animals. The plants include Virginia sneezeweed, which is a nearly endemic plant to the sinkhole pond wetlands. Next slide, please. Now, very quickly, I would like to address conservation. And of course, the most critical issue is that these wetlands, being forested wetlands, have a forest buffer that is retained to protect them. Many people are familiar with a stream management zone. I am forever preaching about a vernal pool management zone, because preserving the critical upland habitat around these wetlands is really preserving the residential habitat of the living community, especially the amphibians which reproduce in these vernal ponds. So that buffering forest surrounding them is absolutely essential to the long-term sustainability of these wetlands. Next, please. And this is based on research that is tried and true. It's been around for 10 to 15 years now. There's plenty of citations for these best practices. And in short, if you look on the far right there, these distances that determine the standard are really based on research into the distances that the indicator or obligate amphibians would disperse away from the wetland. Next, please. Here are some more of the references so that you can dig into these resources. I routinely use them in my attempts to go to bat for vernal pools. Next. So I'm going to wrap up with illustrating here the importance of amphibians to the ecology and their conservation. And I'm going to use one of my old pals, the Jefferson salamander, which is rather common here in the Allegheny Mountains where I am at. And this is a species considered to be of conservation need based on the Virginia Wildlife Action Plan. So here's Jefferson tepals or larvae on the left and a newly emerged salamander on the right. Next slide. Now back to this cool area, this Castile Sinks area. Here are two of the ponds, and these ponds both were identified as having Jefferson salamander populations supported reproductively by those ponds. And then the range, or the buffer circle in red around each, shows the extent to which that particular population of Jefferson salamanders would disperse out from the breeding pond. And this being a complex of wetlands, they overlap, and you realize, oh, here is a polygon, a complex area of the landscape wherein we would need to consider preserving the surrounding forest, the buffering forest. And this is particularly pertinent because this is the national forest where timber production is also a priority for that agency. In truth, the management recommendation here for this site was, keep it all. This area is so unique, such a large complex. Ensure that the entire area around all of these wetlands is preserved to buffer it. Next. Now, very quickly, let me explain why amphibians are so important. We use a technique of estimating relative population sizes around these wetlands to sort of categorize their priority for conservation concerns using egg mass counts. So here's my buddy Hubert, and he's helping me visually count the egg masses of spotted salamanders, which you see here in a site. And that's used to extrapolate this population size estimate. Next, please. I'll show you one quick illustration of how significant this can be. Here is a pretty little vernal pool of excellent quality on top of a low ridge in southern Bath County, the lower portion of the Cowpasture River Valley. And this has 5 obligate species: spotted, marbled, Jefferson salamanders, wood frogs, and the spring fairy shrimp. Nice quality pond. Next slide. Here's what I was particularly interested in. This is a large raft or aggregation of wood frog egg masses. Each mass represents one female or mama wood frog. Now, if you can get there quickly after the females have deposited their eggs and before the water causes them to swell and fuse together, and you can count every individual egg mass, it's a little tedious, then you can extrapolate relative population size. Now, this was nearly 200 egg masses, and that accounts for on the order of 1,000 breeding wood frogs in the surrounding woods that came to this one vernal pool. And what you realize is this wetland supports a living community in the surrounding forest that puts mucho biomass into that system. These animals, whether as adults being eaten by a snake or a hawk, or the eggs or tadpoles being eaten in the pool by aquatic or semi-aquatic animals, they are putting a tremendous amount of food base into that system. And that is very critical to the flow of energy through that system. So I do not lose any sleep at night with the understanding that my favorite group of animals has, as perhaps their most important ecological function, feeding the rest of the world. That is what they do. Next slide, please. Now, finally, and I am done here. I apologize for our challenges. I trust this has come through. I'm wrapping up now so I can take some questions. But it is, of course, important that I mention climate change concerns are really at the center of our studies and our efforts now. The fact that these are seasonal wetlands driven by annual precipitation in their hydrology, and the ways that that is changing now creates an uncertain future for these habitats. You can check out this BBC article linked here. Next, please. Final thought, here's an illustration. This is one of those pools I inventoried for the forest, on the National Forest in Bath County. You'll note at the top, this image, that's the spring, and that's how that pool should look at full pond in spring. This is why we call them spring pools or vernal pools. They are full and teeming with life. Look at the bottom images. Here's what's happening. On the left, that's the same site dry when it should be wet, full of water. Bottom right image, there's the same site when it should be dry and is full of water. I've been seeing this flip-flop in hydrological dynamics in these systems for some years now. And that is what creates the biggest concern for us. Because the hydro period, or length of time that the pool holds water, has everything to do with the biological dynamics and the biological phenomena like the reproduction of these keystone species, frogs and salamanders in the forest. So that's a big unknown. It is pretty disturbing. What do I do to deal with that stress? Next slide, please. I hope there's some chuckles. I keep my head in the wetlands. I stick to my chore of studying these places and monitoring these places. I find my happy place in these wetlands. So I trust this has given you an overview. Go to the last slide, Matt. This is a good time. There is a renewed fascination and interest in these habitats. I want to encourage you to download my friend Stephen's ebook that is brand new, hot off the press. His photography of vernal pool denizens is fantastic. His work was featured this summer in the Nature Conservancy magazine. So do check some of this out. It's very inspiring. And that, of course, is what Stephen does. He uses his photography to inspire conservation of vernal pools. Final slide, questions, please.
Thank you, Mike. We do have time for one question. We want to try and get back on track here. Liz?
Great. What, if any, special measures are afforded to vernal pools by state and federal agencies outside of the usual wetland protective measures?
Gotcha. Legal protection, that's always the big question. In Virginia, we are behind the curve of places like New England and California that have specific state or even local level legislative protection for vernal pools specifically. We do not have specific vernal pool protection laws or laws specifically to protect isolated wetlands. There's been one attempt, but it didn't make it in Virginia. However, the Virginia Department of Environmental Quality has jurisdiction over these wetlands in the state of Virginia, even though these wetlands are isolated, non-navigable, and do not presently fall under U.S. Army Corps or 401 protection. The state does regulate them. No one, under any circumstances, regardless of ownership, can alter any isolated wetland without the express permission through a permit process by the Virginia Department of Environmental Quality. That's the legal protection we have, but it is really incumbent upon us, the citizenry, to be the eyes and ears for this agency and to report, because these are such small scenarios. So to be honest with you, grassroots, progressive, proactive grassroots education about them is probably the most important function right now.
Great, Mike.
Thank you.
Well, appreciate you taking time and being adaptable, as many of those organisms that you talk about have to be nowadays.
My
Talk this
pleasure.
afternoon.
My pleasure. Thank you so much.
Thank you. We're going to take a short 10-minute break. We will reconvene at 2:35 to wrap up our last two speakers.